have the pleasure to introduce Professor Ann-Saada, we have been working many years together when I was at Hadassah time. And we still work together because we are struggling on diagnosis of patients with a lot of biochemical disorders. And it's a pleasure to introduce you and invite you to the stage.
This fantastic meeting. I'm going to talk about mitochondrial diseases, and maybe we can use the patient's fibroblasts to find and evaluate some new treatments. As you can see from this graph, mitochondrial diseases are increasing, and they are not increasing because they were not present. We can now just identify them better, also biochemically and from a molecular point of view. And if I'm talking about mitochondrial diseases, these are diseases which directly affect the OCFOS, and I'm going to talk about inborn diseases due to mutations, and they directly affect the mitochondrial respiratory chain. So the prevalence of these disorders is steadily increasing. It was 1 to 10,000 like 5 years ago. Now it's down to 1 to 5,000 because of the identification of new mutations due to research and also, of course, next generation sequencing. Many of the new genes have been identified by Orly and Professor Arnold Munich, which we heard today. There can be many causes of mitochondrial diseases. They can manifest as a single defect in one single respiratory chain complex, or they could be combined, usually with complex 2 intact. And why is complex 2 intact? Because some of these diseases affect the mitochondrial DNA. And if you can see here, complex 2 is not encoded by the mitochondrial DNA. So we can have mutations in mitochondrial DNA encoded subunits, except from complex 2, mitochondrial tRNA, because mitochondrial DNA is translated by a separate translation system, which is specific for the mitochondria. We can also have mitochondrial DNA disease disorders that we have less of the mitochondrial DNA, reminding you that only 13 of the more than 90 proteins that encode the mitochondrial respiratory chain are encoded in the mitochondrial genome. And that is also the reason why most of the diseases, at least of those we see here in Israel for sure, are encoded in the nuclear genome, because we need at least 1,000 genes to maintain proper OXPHOS, and most of these genes are actually encoded in the nuclear DNA. They could be assembly factors for the mitochondrial respiratory chain. They could be in those subunits. Not only there are 90 subunits, only 13 are encoded in the mitochondrial DNA, and the rest are encoded in the nuclear DNA. We have also all the factors that's needed for translation, like ribosomal proteins and tRNA modifying enzymes, are encoded in the nuclear genome. Also, mitochondrial DNA depletion can be caused by factors that are needed for the maintenance of mitochondrial DNA. We also have secondary effects. We know that, for example, defects in cardiolipin synthesis can cause a combined respiratory chain defect. So apart from those different causes of mitochondrial diseases, we also have a very varied and heterogeneous manifestation. Usually, brain and muscle are affected, which are energy requiring organs, but also the liver can be affected, the kidney can be affected, endocrine system, diabetes, and all sorts of combinations. So I'm not going to mention different names of diseases, MELAS, etc., because there are so many different and new diseases, so you cannot make a real diagnosis based upon clinical observation only. So we used to say that these diseases can manifest at any age, because we also have the heteroplasmic issue. I will not go into that today. Any tissue and any severity. So because of this heterogeneity, clinical trials are limited, and the treatments are mostly symptomatic. These are two review papers, and we see no. We don't have one specific molecule or several that are really effective long term. And in real life, usually these molecules, which could be antioxidants or cofactors, etc., are administered in a cocktail. So you can't evaluate them on an individual basis, and you cannot make controlled clinical double-blind trials only in a very few defined cases. So the whole thing is a mess. So what we need are model systems to model these diseases. And you can have microorganisms, you can have animal models, patient-derived cells. Every model has its own flaws and advantages. But if we're speaking about animal models, for example, you cannot make one animal model for each mutation for each disease, because we are talking about more than 1,000 genes. You can also try to do some work in patient-derived cells. We opted for fibroblasts because they are accessible. The drawback is that the phenotype is not always preserved. You can grow them, but you have a limited. You are limited in terms of generation, and sometimes they're hard to manipulate. They are not cell lines. But this is what we have. This is our model. Some years ago, many years ago now, in 2001, we tested one patient's fibroblast. This patient had a complex 1 disease, the first complex of the respiratory chain. And we saw a positive effect of riboflavin on ATP production from pyruvate, which goes through the first complex. And we were happy with that. But this was only one patient and one compound. And we choose riboflavin because riboflavin is a cofactor. This is a flavoprotein, this complex 1. But what happens if we have many parameters? We have impaired OXPHOS and cellular dysfunction, death, mitophagy, autophagy, whatever. These can be caused by all these mutations that I told you. But the consequences are not only energy depletion. We can have a disruption on membrane potential. Mitochondrial membrane potential is important, for example, for maintain calcium homeostasis, or for importing all those proteins that are needed to maintain the mitochondrial respiratory chain, which are encoded outside. We have ROS production that could cause oxidative damage. We have this vicious circle that if the respiratory chain is decreased, then it produces more ROS. We get more oxidative damage. Then the respiratory chain, as the mitochondrial DNA, takes a hit from these free radicals, and then we have this vicious circle going on. There are those who claim that every one of us will eventually succumb to a mitochondrial disease, which is called aging, because during our lifetime, some of the electrons are escaping from the respiratory chain and they turn to free radicals, and this process is going on. There are some debates about this, but I have to mention it, and it could for sure be aggravating a mitochondrial disease that may present at an older age. You can maybe have some damaged mitochondria and live with that, but if this circle is going on, eventually the cell will succumb. Okay, so what do we want to do? We want to use the patient's fibroblast to see if we can find some small molecules. There are different approaches. I'm only going to talk about small molecules today. So we want eventually to take the patient's fibroblast, put some small molecules on them, and improve OXPHOS and improve cell function. So what do we have? We have antioxidants, we have cofactors and vitamins, because the respiratory chain uses many vitamins and cofactors. We have activators of mitochondrial biogenesis via different pathways, and calcium apoptosis inhibitors, autophagy, etc., modulators. So this is starting to be a bit complex, because if you have all these parameters or all these molecules, what are you going to do with that? And you have a limited passage of the cells. Anna, a former student, and now Dvora and Lisa, PhD students, try to tackle this problem. We have to adapt methods, because not every method is suitable for measuring cells with mitochondrial dysfunction a priori. For example, MTT, XTT assays are based on mitochondrial function, so we cannot use them. So we went to a more primitive method, the methylene blue method. And also we can challenge the cells by growing them without sucrose. If you grow them without glucose, if you grow them in a glucose-free medium with fatty acid from the serum and galactose, they have to actively use the OXPHOS to stay alive. And you can see that after 72 hours, there's a profound difference between respiratory chain deficient cells and normal cells, while in glucose-containing medium, both types of cells grow well. So we use this 72-hour window. If you leave them longer, they will die, to perform most of our experiments. First, as Miguel said, and also you have to determine that the cells are really expressing the phenotype or the defect. So most of those cells we worked with had a profound growth deficiency on galactose. Otherwise, we didn't have a good indication that they were indeed affected. And we tested here, I just gave you some example from the different classes of mitochondrial diseases that we have identified during the years. Most of these cells also had increased ROS, which is expected, but sometimes if you have a very severe defect, the cells are so defect that they cannot even produce ROS. So not every cell had high ROS. And one other important parameter is ATP production, or we can also measure ATP content on galactose, because there you don't have glycolysis. And you can see that most cells have efficient ATP production. This is a summary of some experiments we did on complex 1 deficient cells with different mutations, and we tested different compounds, and we tested 3 different parameters, this was all done in microtiter wells, and we can use 5,000, 3,000 cells per well, and we do triplicates, etc. So this could be done, but then you have to present the results and somehow, and here we try to present them as a heat map. And what we would like to see is to to compare different parameters because, for example, if you have decreased ROS and increased ATP production, that's nice. But if you have, for example, decreased ROS and decreased ATP production, then we're not sure that this compound is so suitable. But I will continue to present our data like this because it's more user-friendly. So here we have, for example, ICAR. We find that ICAR was a favorable compound. It improved growth, decreased ROS production, and also increased ATP production in some complex 1 deficient cells. So we think that ICAR, which in fact is a metabolite of nucleotide metabolism, is working by increasing the mitochondrial content, the mitochondrial biogenesis, and most probably by the AMPK pathway, because AMPK was more phosphorylated. So we think that maybe this patient, who is sadly not alive, but maybe other patients with the same mutation could benefit from ICAR supplementation, and this has also been shown in a mouse model. We also collaborated with Patrick U.Y. Mann from Newcastle, who is an expert on LON, Leber's hereditary optic neuropathy. This is a mitochondrial disease that primarily affects the eyes, one eye first and the other eye comes later on. And it manifests in young adults. And here we evaluated some antioxidants from the ubiquinone family. Why ubiquinone? Because ubiquinone is a cofactor of complex 1. Complex 1 donates electrons to the ubiquinone in the first part of the respiratory chain, and one of these compounds, idebenone, can transverse the blood-brain barrier because the usual substrate CoQ10 does not go through the blood-brain barrier very easily. And we saw that all these compounds could indeed decrease free radicals. But when we analyzed the ATP production, we can see that CoQ1, although it is a cofactor and a substrate of complex 1, and could also go into the brain because it's the short ubiquinone, had a negative effect on ATP production. So this could be some take-home message that not everything that decreases ROS is necessarily beneficial. We can also see that those cells that were benefited mostly from the antioxidants are those who were most affected. The heteroplasmy, this is a mitochondrial DNA mutation, they had high levels of heteroplasmy and low levels of residual enzymatic activity. While those cells who were, had a high residual level and were less affected, benefited less from the therapy. We move on to complex 4, COX-deficient cells, and there we could see that several compounds, mainly ICAR and ascorbate. Ascorbate could donate electrons to complex 4, decreased ROS, and also increased ATP content. So this could be really nice. You can see here that we also tested resveratrol, etc., which decreased ROS, and this could also be an interesting thing. We also saw that ascorbate and increased, and also resveratrol increased oxygen consumption. So this is also a positive thing because they are very bad at oxygen consumption and ATP production, these cells. We did not observe any increase in COX activity, no alteration in membrane potential, no significant increase in mitochondrial content, so we don't know the exact mechanism that these compounds have on the complex 4 deficient cells. Expression analysis, etc., is in progress by Dvora. What we did see, for example, if we look at N-acetylcysteine, which is a good antioxidant, it really decreased ROS content or ROS production, but was negative to ATP content. So here also we have this trade-off effect if we want to use antioxidants. We did the same thing for translation defects, several different defects on the translation system, and I will not go through all these results. We can only see the ATP content, that here also ICAR was good. Bezafibrate is a very good in these cells, while NAC here again, we can see that the ROS maybe went down, but the ATP production also went down. So different cells responded differently, and we have individual results, and what we found is good for one cell, maybe not good for another cell, or even harm. In this line of thought, we also tested some years ago, actually we tried to do a read-through assay, but with gentamicin. That's the story of cystic fibrosis. But we did see instead another phenomenon, that these cells who had a translation deficiency were harmed by gentamicin, where our other cells were not. And these cells are deficient in the mitochondrial ribosome, and we know that translation-targeted antibiotics who target the microbial ribosome, the bacterial ribosome, they have a side effect that they also can target partially mitochondrial ribosomes, which are different from cytoplasmic ribosomes. And if these ribosomes are mutated, they can even harm more. And I will not say anything, but gentamicin is widely used at intensive care units, and if I don't want to open the Pandora's box, but if you try to treat a mitochondrial person with gentamicin and you don't know the exact mutation, this could be a mutation in the translation system, and you can do harm with translation-targeted antibiotics. One of the last examples, this is a secondary defect, a defect in mitochondrial fission. You can see that these mitochondria are long and they cannot divide, and this causes mitochondrial respiratory chain defect. And if you are not convinced by this picture, you can see that we transfected normal cells with a mutant gene, and we can see that the mitochondrial network is destroyed because they cannot even divide. And these cells benefited from bezafibrate and also from idebenone. And this picture just showed that you can go and test different compounds. This is compound X, and you can also test cells that you don't know the exact molecular cause, because this is a personalized medicine. Sometimes you don't know the exact molecular cause or mechanism, but you can still check the cells and see if they respond to a compound or another. For example, these cells benefit from ascorbate. And this is just a summary that the response is individual. Different cells respond differently. You have different responses of different antioxidants. Some cells benefit and some cells do not, and some cells benefit all over the place, and some have mixed response, and some parameters are decreased. And you can see here that ICAR is really bad for these cells, but very good for other cells. So you can actually target the same pathway with different compounds and end up with different results. So the open question remains could the result obtained in fibroblasts, could they be translated into personalized treatment? If we use FDA-approved controls, do we have to go through the animal model? If these compounds are FDA-approved, can we just jump directly from the fibroblasts to the patients? Well, maybe. For at least ascorbat, I don't think vitamin C can do no specific harm. We hope to continue to this work, and this work was mainly done by Dvora and Lisa. And I thank Corinne, who worked with me with the diagnosis of most diseases. Molecular diagnosis was done by Orly, and we collaborate with many treating physicians. Thank you.
Any questions? Yeah? One question.
Just a quick question. Have you also looked at the ATP to ADP ratio? Or only
no,
total?
but we have looked at ATP production versus ATP content and on different medium, and they can differ. And we have to elaborate that later on. Okay?
Thank you very much.